Now students, in today's lecture, we are going to study properties of RBCs that are erythrocytes or red blood cells. Now, what are these properties? First property is Rbcs formation. So, when the blood is taken out of the blood vessels, the RBCs pile up. So this piling property of RBCs, this property of the RBCs to make the pile is actually the Rbcs formation. So this property is actually accelerated by the plasma proteins that are globulin and the fibrinogen. Now, second property is the specific gravity. The specific gravity of RBC is 1.092 to 1.101. Now, what is packed cell volume? We have already discussed in our previous lecture that packed cell volume, that is abbreviated by PCB, is the portion of the blood that is occupied by the RBCs or the red blood cells and expressed in percentages. That is the portion of the RBCs that is in the hematocrit tube, that is at the lower level of the tube, that is at the bottom of the tube. So, this hematocrit value is expressed in percentage. Normally, it is 45%. Now, coming towards the stability of the RBCs, that is suspension stability. So, during circulation, these RBCs remain suspended uniformly in the blood. So this suspension ability of RBCs is actually the suspension stability. We have heart, which is working in our body, that is continuously pumping the blood. So because of this property, our RBCs are continuously suspended in our body. Our RBCs have negative charge in our body. So they are not attracting to one another. They are not making the pile and they are continuously moving in our circulatory system. Now, what is the lifespan of RBCs? The lifespan of RBCs is 120 days. And after the time, now the cells will be over or the senile. And these RBCs are now get destroyed in the reticuloendothelial system. That are, these cells will be undergo phagocytosis and will undergo destruction. And ultimately, after the destruction of the RBCs, there will be release of hemoglobin. So this was all about the properties of RBCs.